All right, welcome to a new video. This is going to be a tutorial of using the website Geneshot that we developed. Geneshot, in a very short, brief explanation, is a website that can do very fast literature lookup of any arbitrary search term that you give it, and it will retrieve gene symbols that match these publications associated to your search term. And then from that, you can use those genes that you retrieved to identify genes that are not previously associated with the search term that you're looking for. And it's doing that by using a couple of different gene-gene similarity matrices. These matrices can come from different sources, and right now Geneshot supports three types. The first type is coexpression using RNA-seq data. Derived from the ARCHES4 dataset. And then as a second type of similarity matrix, we can use the co-occurrence of genes in publications. And for those, we have three datasets. This is the TAGR dataset, the AutoRIF dataset, and the GeneRIF dataset. And then as a third type, we have something called the enricher co-occurrence. So I will go through the website in a couple of steps, and I hope this is a useful video for you. This is the starting website of Geneshot. And what you can see here is that we have two fields. We have search terms, and then we have not search terms. So you can specify things that you don't want to look at. So we keep it simple, and we just search for one of the examples that we put in here, but in this field you can type anything you want. So let's search for genes that are involved in glioblastoma. So if I click on this, it is equivalent of just typing glioblastoma in here. And what you can see here is that we have a selection between GeneRIF and AutoRIF. So these two databases are very related to each other. And what they do is, we did a previous literature review where we scan all the abstracts from all the publications, and we look for gene symbols in those abstracts. And so from this, we know what genes are associated to a publication. This is AutoRIF. GeneRIF is a manually curated resource. And so there's people going through the abstracts and through papers, and they identify if a gene is in a publication or not. So GeneRIF and AutoRIF, they're basically a dictionary that maps PubMed IDs to gene symbols. So when we do this search, in the background of the server, we do a PubMed query and we identify all the publications matching this term. So if you want to specify this more, you could technically add filters that you do not want to use in this analysis. But so we have run analysis, and you saw like it went pretty quickly. And what we see here is this scatter plot. And each point in the scatter plot represents a gene that matched to that search term. And we know that the gene matched this search because there were publications citing this gene. So if we go, for example, to this gene, this is MGMT, and if I click on this, we actually see that there is a histogram showing the trend over time. So in purple, you can see the numbers of citations of this gene that are not associated to the search term, and then in blue, you see the publications associated to the search term. So this is quite interesting because what we can see is that in around 2006 or 2008, people started to associating MGMT with glioblastoma. And at this point, half of the publications that cite this gene are also associated to glioblastoma. And so one important thing to remember is that some genes are just generally highly referenced by a lot of papers, and they're not very specific to the search term that we applied. there would be an example like EGFR. EGFR is a very important protein or gene, and it will be associated to a lot of other factors. So if we look at biological processes, so if we look at this histogram, we see that the fraction of publications that are specific to glioblastoma is fairly small in the general context of all the publications. So it's still a highly cited paper. So there is 1,140 publications that cite this gene, but it is still a small fraction of the total publications citing this gene. MGMT, on the other hand, also has a lot of publications, but also the fraction of publications that are also matching glioblastoma is fairly high. So this is a more specific gene. It's more specific to the search term that we applied. And this is also true for this gene. Here we have IDH1. IDH1 also has a fairly high specificity, so about 30% of the publications are associated to glioblastoma. These, on the other hand, are very specific, but they have very few citations in general. So this gene here, for example, has only one citation, and that citation is associated to glioblastoma. So now we scroll down, and what we see here on the bottom, this table, is the same representation as the scatter plot. We have the gene symbol, and then we have the total publication count that matched our search term and cites this gene. And then we have a fraction of this number divided by the total number of publications of that gene. So even though there's more than 1,000 citations for EGFR, this is only 5% of the total publications citing EGFR. But we can immediately see that some genes are more specific. Here we have a list of interesting families of genes. We have kinases, GPCRs, and ion channels. And these are of specific interest because they're easily druggable and they might present interesting drug targets for further study. And each family has also this dark category. And the dark category that comes from the Illuminating the Druggable Genome project, where researchers identified genes from these three families that have very few citations, very few, very little knowledge about them. So these are doubly interesting because they're potentially targetable by a drug. And nobody really knows in what context these genes should be targeted. Now comes the prediction step of Geneshot. So on the side here, we have genes that are predicted to be associated to glioblastoma. And while these genes might already be known to associate to glioblastoma, the algorithm is using these predictor genes on this side and identify genes that are similar to this set of genes. And it basically associates these genes now with glioblastoma indirectly through already known contributors to this context. And how does it do this, right? So here, up here, we basically rank the top genes, in this case 50 genes, and now we calculate the average correlation of all the genes to those 50 identified genes that we have from our query. In this set, we did the enricher co-occurrence, but we can also go with ArchIS4 coexpression. So if I recalculate the step, this table gets refreshed and we get a different set of genes. And I can also do this with the literature-based similarity matrices. So here we have autoref, which means we want to look for genes that co-occur in the abstract with the list of genes that we found before. So I can recalculate the matrix, and this is calculated on the fly, and it's pretty fast. So we can see there's actually IDH1 So as I mentioned before, this prediction does not discriminate against genes that are already known to be associated with glioblastoma. And as I said before, IDH1 we already found through the literature search. And because we used a literature search again, these predictions came back up. The same is true for MGMT, but there's still some genes in there that were not associated with glioblastoma. If two genes co-occur in publications not associated with glioblastoma, then a novel gene can be predicted for this search term. So then we have some simple functions here. You can click on this button and that will submit these genes to Enricher. So if I click on this, we get the results. So you can see we have 200 genes associated or predicted to be associated to glioblastoma. And then we have some potentially interesting commonalities of these genes. We can already see in the wiki pathways that the number one search term is signaling pathways in glioblastoma. Not that surprising that that worked. So this is the main functionality of this website. It's very simple. It's supposed to be simple, and it allows you to very quickly get an idea of the field or any search term and what genes might be interesting. So it's just like a piece in the puzzle. If you want to identify some interesting gene, or you already have an idea of a gene and you need some confidence that this is a good target to follow up on. So the website has some other functions that I want to show. So we just looked at the PubMed query. The next one is the gene function prediction. So if we go here, we get to a very similar view. And what it allows us to do is we can enter any gene symbol that we want, and we will get through our algorithm a prediction for certain libraries. So here we have certain categories that we can predict. So we can, for example, predict potential phenotype associations, biological functions through gene ontology, and KEGG pathway memberships, kinase interactions, transcription factor, upstream transcription factors, things like this. So if we go here and we want to search for a gene, I have to think of something on the fly. So we looked at this gene before, right? So let's just try it and see what happens. So here we want to use the, let's say we want to use the coexpression for Enricher. So if I send this to submit, we get a result of predictions. And as we can see here, there isn't really too much known about this gene for the mammalian phenotypes. So usually we see an AOC on this side if there's already some information about it. And it will show us how well we recapitulated the known biology for this gene. But these are the predictions. So this is just one example. And you can look at the paper and see at how well these predictions actually work. And they generally work pretty well. This is not universally true for all genes, but on average, especially things like biological processes can be identified fairly well. So in this case, it's not phenomenal. The AUC is 0.06, but they can be potentially interesting things here. So for example, here's one example where we can click here. This is TNF, and we predict the biological process, and we used Enricher co-occurrence. And then, so here it's already better. The AOC is 0.73. So we could recapitulate, the function fairly well. And we have some very strong predictions. So these z-scores, they're kind of z-scores, they're like fairly high. So these are interesting functions that we predict. Now, the next category is called gene set augmentation. And it can give us an idea. If we put a list of genes in here, it gives us an idea of how common those genes are. And we can also calculate how, what the genes are that are associated to this. So here again, we have the different similarity matrices. And in this example, here I have like a preset set of 3 gene sets. One is very common genes. And as we can see, all the genes, there's 20 genes, they have a lot of citations. So we can see here there's a lot of publications for these genes. But if I go to the mixed, so now I have a more evenly spread publication count for those genes. So we can immediately see the most cited gene is this one. And then there's also some genes that don't really have a lot of citations down here. But again, we can calculate the similarity of all the genes to the similarity of these genes given a pre-selected context. So this, is an interesting tool if you already have a gene set and you might want to find something that's related to this even more. And then, if you want to use this programmatically, you can. So this whole website is based and built around an API. So if you go to the API section up here, you can learn about how to write your own code to get the results programmatically, and you can build it into your program and send much more queries than you could do by hand. So I hope this helps. It's a very brief introduction to the website. If you have questions, feel free to send us emails. We try to be responsive. And I hope this is useful for your research. And see you next time when it's the next video or when you send us an email. All right, bye.